This is a tutorial of the DRsc single-cell RNA-seq database. I'll be giving an overview as well as showing a few example use cases. The DRscDB was built from the curation of 47 publications of fly, human, mouse, zebrafish, and mosquito, and both cluster-level gene expression when available and marker gene annotation were captured and stored in our database for mining. For a summary page of all the datasets, you can click this link or the Datasets tab. And this table shows an overview of all the datasets that we have available. You have link outs to the PubMed IDs as well as the publication itself, and then a variety of metadata about each particular publication. We also have the full list of marker genes available. If you click on the Markers tab, you can see a summary table here with the publications, again, link outs, species, things like this. And then if you click on the details for a particular publication, you can see the marker genes and the clusters. Clicking on the details gives a detailed view of each particular cluster. And then with all of these tables, you have the option to sort by clicking on the column header. You can also search the tables directly or export to either Excel or CSV. So the first use case I'll be showing is a single gene search. You'd start by choosing the species, entering the gene name, and then choosing a filter, tissue to filter by. You also have the option to filter the ortholog results. For now, I'm just going to use the best match only. This takes you to this ortholog results page. The search term is highlighted in blue. You can see the other species that results were brought in for, as well as some link outs again. This column gives the ortholog mapping score. If you're looking for more results, you can either adjust the filter or entirely remove it. And then clicking this button will update these results, and as you can see, there's now far more. This example, I'm just going to keep the best match. And then on the far right, this shows the number of single-cell RNA-seq datasets in our database. Clicking the results takes you to a overview page summarizing all the single-cell RNA-seq datasets we have. There are link outs to each particular publication, but basically we give the dataset name, the tissue type, the clusters. If you actually click on the clusters link, it'll again take you to page for marker genes and again more detailed information for each particular cluster. Scrolling to the bottom of this page, there are two links for cluster level statistics and another for marker statistics. Starting with the cluster statistics, you're taken to this page with, by default, showing dot plots for each publication. For example, with this one, users can compare by different age groups, and then the dots themselves represent the percent the cell is expressing the input gene, and the darkness reflects the expression level. Scrolling to the bottom, there's a summary table of the cluster level statistics. Again, with all these tables, clicking the header allows you to sort them. You can search them directly, and then if you click on the dataset IDs, you can filter this way as well. And then if you want to export, you can either get the file in Excel or CSV format. Going back to the top, you also have the option to view these results as either bar graphs or heat maps. And then with the heat maps, you actually have the option to customize them a bit, so you can change this to average expression. You can change the color of the heat map. And then with all of these figures, if you wanted to export one, you can click this button here and save it as either an SVG or a PNG file. Going back to the summary results, now I'm going to show the marker statistics. And with these graphs, the height of the bar is showing the average log fold change, and the darkness reflects the p-value. Again, at the bottom, there is a summary table of the marker statistics. Like I said, sortable, searchable, and you can export. So that pretty much covers the single gene search. Next is the single gene list enrichment search. So you would click on the Enrichment tab, and then this kind of search would be useful for users who want to analyze a list of genes, for example, hits from a cell-based screen, or a list of transcription factors in order to find out the tissue or cell types that are enriched. This could be useful if, for example, you're trying to identify the most relevant tissue or cell type from an input list of hits from a cell-based screen, for example, some fitness genes, and this would allow the user to design an in vivo experiment accordingly as the next step. Users might also use a list of genes from a particular group, for example, transcription factors, in order to identify the transcription factors that are expressing in the tissue or cell type of interest for a follow-up study. But before we do the search, because the enrichment is very computationally intensive, we basically separated the process of gene ID mapping into a different step. So you'd first click on the Gene ID Mapping tab, choose the species, and this basically maps your gene IDs or gene symbols to the same version that we're using. So you would paste your gene list here, click Submit, and it will show you basically your search term and the gene symbol that it was mapped to. So going back to the enrichment page, I've went ahead and already done this, so I'm going to paste my gene list. I've already got the species and gene identifier selected. You have the option to choose the number of top marker genes. For this example, I'm just going to keep it at the default of 100. And then you have the option to filter by enrichment gene sets. You can choose species, tissue, and then a variety of specific data sets from publications. For this first example, though, I'm just going to show all. So after clicking Submit, you're taken to this enrichment results page. You have the gene set names, the percentage of the genes in the gene set, the fold enrichment, the p-value, a variety of information there. You also can choose to either show or hide some of these columns. Scrolling down, we have some visualizations. There's a few default options. You can click through these to see these figures. But if you'd prefer to customize them yourself, you can also directly set things like what the bar represents, what the color represents, and then what actual color scheme, and then clicking this will update the graph. Again, also if you hover over each of these bars, it'll tell you the exact value and the exact information. If you want to see again which gene list you are using, you can see here, and that pretty much covers the single gene list enrichment. So now going back to the enrichment page, now I'll be showing an example of a multiple gene list enrichment search. So from the enrichment page, you would click this button, and like the name suggests, this type of search allows users to analyze multiple gene lists together. For example, marker genes of each cluster from a newly obtained single-cell RNA-seq dataset that could be compared with a relevant dataset and used to assign cell types based on the existing dataset. For this example, I'll be using the marker genes of blood cells from a single-cell survey of Drosophila blood, and I'm going to go ahead and paste those in now. So the format for the marker genes is the group ID followed by the gene, and you would separate it with a comma. You can either use gene symbols, species-specific gene ID, or the entree gene ID. And then I'll be comparing these with the markers from a similar study, single-cell transcriptome maps of myeloid blood cell lineages in Drosophila, which is this dataset. And users can do this kind of comparison to validate data reproducibility. In addition, if you're doing this type of analysis with a dataset from a different species, the results can facilitate the discovery of evolutionarily conserved transcriptomic architecture. And I'm going to show first the top 10, but also the top 100 markers to compare side by side. And we do suggest not using the top 10 markers for cross-species data comparison, since not all the marker genes are conserved. Alright, so starting with the top 10, Go ahead and submit. So this is going to take you to this results page. First you have a table showing the p-values and fold change for each cluster. You can click on each cluster to see detailed information, as well as generate figures similarly to what I showed before. Going back though, you have the option to generate heat maps or a dot plot. With the dot plot, you do need to select Generate Graph first, but with this one you can again customize it, so it'll show potentially different colors or change what each thing represents. You have some other customization options where you can essentially change the order of the clusters and basically just change what both axes shows. So you can use this to rearrange these. And this will update here after clicking generate/refresh. And then for comparison, you can also show the top 100. So paste this again. And then you can imagine you can do a variety of different comparisons for different data sets and a lot of different use cases. And so finally, I just want to mention that we encourage users to submit their own annotations. We have template files and a few examples. You'd fill out this form and then submit your own annotation there. For more information about how to use the tool, you can also visit the About page, where we have more detailed instructions. And then if you have specific questions, you can either email us directly here or use the software bug report form at the bottom of the DRC trip homepage, which is available here. And thanks for watching.